Hello, I am Vivek Ruhela from SBA Lab, IIIT Delhi. I work in genomics under the supervision of Dr. Anubha Gupta and Dr. Ritu Gupta. Here I am presenting my work miRPipe, which is the Unified Computational Framework for robust, reliable, and reproducible identification of novel miRNA from RNA-seq data. So here I would like to emphasize 4 important points of miRPipe. That is, what is the essential idea behind miRPipe? What are the gaps we are trying to bridge? How miRPipe works, and the benchmarking of miRPipe. So let's check the essential idea behind miRPipe. In human and non-human organisms, the miRNA regulates a plethora of cellular level mechanisms such as cellular metabolisms, regulation of biological network and pathways, etc. Also in many diseases, especially in cancer, miRNA have been classified as a potential biomarker that influences several transcripts or a network of oncogenes, tumor suppressor genes with distinct functions. So our aim is to provide such a unified framework that can identify miRNA accurately and reliably from NGS data. Now the question that arises here is that what are the gaps we are trying to bridge using miRPipe? There are several existing state-of-the-art pipelines in the literature. For example, miRDiB2, miRDiB*, miRNovo, miRPro, sRNA toolbox, etc. Each of these pipelines has some pros and cons. For example, some pipelines are very fast and optimized. Some pipelines can identify RNAs from several genomes. Some pipelines can identify multiple types of RNAs other than miRNAs, etc. But still, there is an open challenge, which is about the pipeline accuracy. And also, many of these existing pipelines do not provide information about paralogues and miRNA belonging to the same phylogenetic tree. I will talk about paralogues and miRNA in the same phylogenetic tree sooner. So here we are trying to fill these gaps by developing a pipeline miRPipe such that it can not only identify miRNA accurately, but also provides many other pieces of useful information from RNA-seq data. So now let's talk about how miRPipe works. So the workflow of miRPipe is shown in the figure 1a. So miRPipe takes a FASTQ file as an input, and the first step of miRPipe is the data preprocessing. The data preprocessing contains 2 sub-steps, that is adapter trimming and the length filtration. The adapter trimming steps remove the adapter primer sequence from the FASTQ file, and in the second sub-step, length filtration, the FASTQ file is split into 3 parts, where the first part contains the sequences of length in between 17 to 24 nucleotide. Second part contains the sequence of length 25 to 31 nucleotide, and third part contains the sequence of length greater than 31 nucleotides. After length filtrations, the data preprocessing step is completed, and then pipeline is split into 2 sub-pipelines. First one is the pi-RNA identification pipeline, and second one is the miRNA identification pipelines. Both these pipelines run simultaneously in a separate thread. In the pi-RNA identification pipeline, first step is the quality filtration, where we filter out all the sequence that contains more than 2 nucleotide of FRET quality score less than 20. After quality filtration, we map those filtered sequence with the human genome using type 1 with a condition of 0 mismatch in their seed region. After that, we annotate those map sequence with the pi-RNA database and then perform the differential expression analysis using DSeq2 to obtain the deregulated pi-RNAs. On the other hand, in the miRNA identification pipeline, first the FASTQ file is mapped with miRDiB* to identify the known and novel miRNAs present in the FASTQ file. So please note that in a miRNA identification pipeline, we take the FASTQ file of sequence 17 to 24 nucleotide length, and in the pi-RNA identification pipeline, we take the FASTQ file of sequence length 25 to 31 nucleotide length. So let's come back to the miRNA identification pipeline. First, we map the FASTQ file with a miRDiB*, and then we do some post-processing of those miRDiB* output to make it more readable. And after that, we take all the novel miRNAs identified by miRDiB* to the -R database for blast search. The reason behind it is that in many scenarios, if there is a reverse complement of the non-miRNA sequence present in the FASTA file, then miRPipe annotate that reverse complement sequence as a novel miRNA, which should not be annotated as a novel miRNA instead. So here we take all the novel miRNA sequence, take it to the DASHA database, thus search it, and if DASHA database tells that this sequence belongs to any known miRNA, then we correct it again. So after blast search, we do the novel seed-based clustering using CD-HEAT, which is the heart of the pipeline. So novel seed-based clustering is shown in the figure 1b. Before this, discussing about novel seed-based clustering, I would like to discuss about the seed and exceed region of the sequence. So in any miRNA sequence, there are two parts, two types of parts in miRNA sequence. The first one is the seed region part, and second is the exceed region part. In most of the cellular level mechanisms, the seed region plays the most important part. So in novel seed-based clustering, there are three possible scenarios. The first one is when there are zero mismatch in the seed region, and when there are up to two mismatch in the exceed region, and their mapped genomic location is same. In that case, the expression counts of two novel miRNAs are merged. In the second scenario, if there are zero mismatch in the exceed region, if there are zero mismatch in the seed region, there are up to two mismatch in the exceed region, and their mapped genomic location is different. In that case, these two novel miRNAs are called as paralogues. And in the third scenario, if there are zero mismatch in the seed region, more than two nucleotide mismatch in their exceed region, and their mapped genomic locations are different. In that case, these two novel miRNAs are called as the miRNAs belongs to the same phylogenetic tree because they share the same seed. The expression count in the second and third case are not merged because these two because their mapped genomic locations are different. After checking all the novel MIRs using novel seed-based clustering, we compiled all the updated expression counts of all the samples of all the known and novel miRNAs into a single file, and then perform this differential expression analysis to identify the deregulated miRNAs. Once we have the deregulated miRNAs from DC2, we can get the deregulated miRNAs and deregulated pi-RNAs from the data, and then we can do a further analysis like miRNA gene interaction, miRNA disease interactions, or networking analysis, etc. So this is the workflow of the miRNA pipeline and novel seed-based clustering. After that, we have performed the benchmarking of the miRPipe against the 7 available state-of-the-art pipelines. Here, what we have done that we have performed the benchmarking with two types of experiment. First one is the synthetic data experiment, and second one is the real-time clinical data experiment. So synthetic data experiments are performed using our in-house tool miRSIM with 3 read depth categories. Here, we have generated the synthetic data using our in-house tool miRSIM in 3 read depth categories. First category of data contains the synthetic data of read depth 10,000. Second data contains the FASTA file, synthetic FASTA file of read depth 1 million. Similarly, the third category of synthetic data contains FASTA file of read depth 0.1 million. So here, figure 2 shows the benchmarking results of all the synthetic data experiments. So here, we can see that figure 2a shows the benchmarking results for the pi-RNA data for pi-RNA experiments. Figure 2b shows the novel miRNA benchmarking results using synthetic data experiment. Figure 2c shows the known miRNA benchmarking results using synthetic data experiment, and the figure D is represent the overall pipeline assessment using synthetic data experiment. So here we can see that miRPipe is outperforming in all these 4 cases. In case of pi-RNAs, in case of novel miRNAs, in case of non-miRNAs, and then overall. After that, we have checked our pipeline using real-time CLL data, and then we have observed that our pipeline is giving the least false positive and false negative as comparison to the other pipeline. And also our pipeline was identifying the most number of the reported miRNAs and pi-RNAs in the real-time clinical data. So here we have used the clinical data of CLL from one of our previous publication in Blood Cancer Nature journal. Once we have done our pipeline, we have encapsulated our pipeline in Docker so that it becomes the platform independent and anyone can use it. The advantage of encapsulating miRPipe in Docker is that the user doesn't have to worry about installing all the individual dependencies or OS or library conflicts, etc. User has to download the Docker image and run it directly. Currently, this work is under review and sooner it will be published. In the end, I would like to thanks to my supervisor, Dr. Anubha Gupta and Dr. Ritu Gupta. Also, I would like to thanks Dr. Sriram Krishnamachari and Dr. Gaurav Ahuja from CB department and Dr. Gurvinder Kaur from AIMS for their wonderful support and guidance. In case of any further query, please contact me. Thank you.